Springhouse, US. She studied her PhD in biochemistry in 2006 from the Institute for Genomics and Integrative Biology in India. And then she moved to Germany to do her first postdoc with the Professor Axel Ulrich and then Dr. Henrik Daub at the Max Planck Institute of Biochemistry. And then she developed a highly innovative chemical proteomics approach that delivered drug affinity data. And then her patented technology is commercialized through the company, Evotech. And in 2010, she joined the lab of Matthias Mann, Professor Dr. Matthias Mann. And since the beginning of this year, she is working as a principal scientist and group leader at Janssen Janssen. Now she's going to tell us about the computational PTM proteomics.
Thank you. Hi, everyone. Thank you for the kind introduction and the invitation to be in this summer school, first time as an external speaker, but it still feels like coming back home. And what I would be talking about today is most of the work that I did at, in Matthias's lab and working together with Jürgen. It had been fun, and I will be happy to share that journey with all of you. So as this talk is placed right after you are all now familiar with how to work with MaxQuant and Rometa scores out there for protein expression abundance. Today we have two sessions, one on the PTMs, and after me it'll be Marco who will be talking more about the interaction proteomics. So today we will give you one lecture followed by a tutorial, first for PTMs and then for the interactomics. So that's how this session is organized. And the first part, of course, lecture is going to be more theory, and I'll try to keep it a balance of giving you details yet trying to still give an overview. So as a biologist who had never touched mass spec during her PhD, but was always fascinated by how cells relay information, what are the signaling events out there, how does cell know that now I have to die or I have to replicate? All those decisions are somewhere encoded in the signal relay information there. And I am particularly fascinated by how PTMs play a role in this whole pathway. So if I really break it down to what are the fundamental questions in the PTM world, the one very basic question would be what fraction of the proteome is modified? And how much ever simple it looks, I have to say, before I started my postdoc and after I finished as a project leader, it changed completely thanks to phosphoproteomics. We'll talk about it. The next was, if we know what fraction of the proteome is modified, which proteins individually are modified, and if we know which of them, what particular sites on these proteins are modified. So we are going like zooming into one layer each. Next question is, okay, there are so many of them. Are these sites changing in the biological process of my own interest? Are they changing during differentiation, cancer, drug response, whatever is your area of interest and whatever is your area of modification that you like to work on? Next question is, even though I understand that these are the sites that are changing, what is the fraction, that is stoichiometry, of these modifications that are changing? So is it a percent of the protein that was modified and now it's become 10%, or was it 10% to begin with and it's now 90% in response to the stimulus? So these are two completely different things compared to the fold change. We'll talk about stoichiometries later during the talk. And now we get one step further. Do we know the enzymes that modify these sites? And if we know all of this, do we know if there is any cellular function for the modifications that we are working around? While the first four questions here have seen a lot of benefit from the advances in phosphoproteomics and similar fields, I would say the last three still remain challenging. Challenging when you talk about them on global scale. So this brings me to really the aims of phosphoproteomics, which are essentially addressing those questions. But when you talk about the questions which are more focusing on the functional role of these sites, I think it has to complement with the other methodologies to bring the whole field together. Now, again, as I mentioned, that there have been a lot of advances in the field of PTM proteomics. I'll make a disclaimer here. For me, PTM is equal to phospho, so please change it with your favorite modification in your mind as I speak. The concepts are same. Again, so since most of the modifications are sub-stoichiometric in nature, enrichment is an inevitable step right now, but we hope that with better instruments, we'll probably reach a stage when we don't need to enrich these modifications. So that will be something we can look forward to. So a great deal of work has now gone in bringing new tools for enriching modifications. Phospho has already been there on board, but for the other modifications, there have been new antibodies that have come on board and have benefited deep analysis of those modifications. However, I would say a big chunk of advance comes from the tools, computational tools. That's why I think all of us are here in this summer school. The PTM spectra are much more complicated than a regular peptide spectrum. And accounting for all of that in a good search engine, that is a completely different level of sophistication that is required. And thanks to Jürgen and the team that we are doing great advances here, which have helped us to get the depths that we want with the robustness that should come together with these platforms. And lastly, all the advances on quantifications have brought us to a stage where we can do PTM proteomics label-free, and that's what I'll also try to convince you during this talk, so much so that we can calculate occupancies from label-free data. That is the most challenging task, I would say, that has been addressed with label-free data sets. So while enrichment methods are outside the scope of this talk, because I would never reach then where I want to reach with this talk, but here is a paper that I would highly encourage you to read by Josh Kuhn's lab, which talks about the top 30 studies in the recent times and kind of clusters the methods of enrichment, fractionation, and quantitation used by these major studies. It's actually a table of 30 different studies, but just to give you a glimpse, I've cut the table into half. You would see that there is a very clear shift from SILAC to label-free for a lot of these large-scale studies. That has happened because we now have robust chromatographies, instruments and the normalization and computational methods that are around. The other thing is that while a lot of depth can be obtained with fractionating the proteomes or phosphoproteomes, there is a good trend on moving towards single-shot analysis that increases the throughput of our experimental designs. And for phospho, there have been these major methods out there already, titanium, IMAC, or similar. But for new, there are some antibody-based methods for tyrosines, and there's been jury out there which of them is best or not. So just to say that it is not uncommon now to work with tens of thousands of sites and even getting a lot of them within single shots. So the data set that you will be working later today with the tutorial, and where we've also described a great deal of computational details, the computational PTM pipeline in MaxQuant is this particular paper. This was first paper where we showed that label-free methods work for phosphoproteomics. And the other paper that I would encourage you to read from one lab is paper from Sean, which showed how you can increase the throughput to get a reasonable, pretty good depth in single shots. So again, I'm going to switch in between in my mind as a biologist and a phosphoproteomics person every now and then. It'll be reflected in my talk also. So again, this particular paper gives you very, very good details on the computational pipeline employed in MaxQuant for phosphoproteomics analysis. Second, that this particular project was very intriguing for me, that if you just focus on one cell line, 3 stimuli, what is the maximum number of phosphopeptides can you see? And this was amazing to me to figure out that it's much bigger than I was thinking. It's 50,000 peptides in one system in 3 conditions. And think about the whole human body and different organisms. Where are we going with this? The other important aspect of the study was that label-free data can now be, label-free quantification of phosphopeptides can be done reliably, so much so that you can get the occupancy estimates from there. And here I would like to say that while I have quoted this particular estimate many times myself, I am assuming you're as guilty as me of saying that 1/3 of proteome is phosphorylated. No. We have evidence for at least 3/4 of proteome to be phosphorylated. And I use at least here very carefully because I know it's growing much more. So this brings me to the outline of the talk today. good data, good analysis. So we first need to get good instruments out there, break your PTM peptides in a way that you can get information-rich spectra out there. So that's why a little bit of introduction into fragmentation methods and focusing on phosphopeptides there. I'll tell you that what are the advantages and disadvantages of 3 major fragmentation methods. Second, we'll talk about how the information that is now generated at the end of your mass spec analysis is dealt by in MaxQuant, which ions are considered to calculate Andromeda score, how is it different from normal normal proteome search. Then what is very exclusive to PTM searches, you have to go ahead and calculate, identify the site of localization after you've identified the peptide. And next are, you do calculate the fold changes, but you can also go ahead and calculate the occupancies. How is it done? I hope I can explain it to you in the simplest way. And lastly, talking about some of the practical aspects of how would you set up a PTM search in MaxQuant. And if there is some time remaining, then we'll talk about tutorial workflow, which is going to happen after the lunch. So first, the very basic, before we get into fragmentation methods, here is the fragmentation nomenclature. If you're reading peptide breakage, depending on where you break the bond, from N terminus, you call them ABC, or from the C terminus, you call them XYZ, and these numbers represent the number of amino acid side chains that are included in this ion. With this, you can now focus on 3 different fragmentation methods that are out there, which are most commonly used for phosphoproteomic analysis or other PTMs. Collision-induced dissociation, CID, HCD, higher energy collisional dissociation, and ETD. So basically, while CID and HCD, they would break this CO and H bond, they will mainly produce the B and Y ions. So here, the Y and the B ion, whereas the ETD, which mainly works by breaking the NC alpha bond, would produce mainly the C and Z ions. And now getting specifically into individual fragmentation methods, what are the pros and cons of them? CID, which has been the most conventionally used traditional fragmentation method, the advantage here is that you get almost 100% conversion of your precursor to fragment ions. And you have this MS to the nth capability. By that I mean MS, you isolate the ion you want, you do MS, you can go ahead and so on and so forth. However, the disadvantages are that you only trap the larger fragment ions, so the so-called 1/3 cutoff rule. And this is a little bit annoying, if I may say, for phosphotyrosine analysis, for example, which produces a very small diagnostic peak, which is used to confirm that this was indeed a phosphotyrosine peptide. I'll come to that in a couple of slides. And the other big problem is that CID follows the lowest energy path of fragmentation. By that, what I mean is that if there is a labile PTM, that's the first thing that will get off your peptide before you even break the peptide. So that's not where you want to go if you want to generate an information-rich spectra and you want to know where is the PTM located on your peptide. So how is this in real practical world? It's really a huge problem when you talk about the phosphoserines. You see that when you do MS fragmentation for this phosphoserine containing peptide, you actually see only almost 1 peak, and you do not see other fragment ions which you would need to get the sequence information. However, this is little less of a problem for threonine and actually not at all a problem for tyrosines, where this ring structure of tyrosine stabilizes this whole structure, and what you instead get is formation of this ammonium ion, which, as I was referring to a couple of slides ago, can be the diagnostic peak. So it adds more to your confidence in identifying the phosphotyrosine. But as we all know, in a typical phosphoproteome experiment, most of the sites that you see are serine or threonine. So this problem had to be addressed. Back then, there were 2 different approaches that were used to deal with this neutral loss. What we call as MS3, since CID allows you to MS to the nth capability. What you do is you fragment further the neutral loss peak. By that, what I mean is, if you have, for example, this MS2 peak, you take the neutral loss peak, you fragment it further. So compared to this top spectrum, the bottom spectrum has additional fragment ions which can be used to get the sequence information that you were missing in the scans before. However, it comes at a cost. Instead of doing one scan event, you're now doing 2 scan events per phosphopeptide, right? Which means you're going to get half the identifications than what you would have gotten before. And to address this, Thermo came up with a very nice idea, which they called as multi-stage activation. So here you do not wait for a neutral loss, but what you say, you assume that there will be a neutral loss, and you already call for an additional activation of neutral loss peaks. So the result of that is that you get your MS2 and MS3 spectra combined in one pseudo MS3 spectra, or what you call as multi-stage activation put together. So you're back to the same time dedicated to one phosphopeptide in your analysis. So you do not compromise on the depth per se. With this, in 2010, with the Orbitrap Velos coming in the market, there was option to also test HCD as another fragmentation method. So basically, what you could do now is you could fragment your peptides in the HCD cell, but what was really cool was now you could measure your fragment peptides also in Orbitrap. So you get the ppm accuracy not only for the parent ion, but also for your fragment ions. And that's really good if you think about now with higher accuracy, it's much easier, or it's much more favorable for database searches downstream, right? And the other important thing was that you do not see this 1/3 cutoff rule, which means you're not going to miss any information on the lower mass ions, which would be very favorable for detecting, for example, the ammonium ion for the phosphotyrosine. So that was really good. So you get 2 benefits again. High mass accuracy because you're measuring everything in Orbitrap cell. And the second one is you do not miss on the diagnostic peak that comes with phosphotyrosine. Now, coming to ETD, which has become popular in the recent times, but is mostly used in combination with CID and HCD. So the advantage of ETD method specifically is that unlike CID or HCD, where the labile PTMs are the first ones that will go off your peptide, ETD is actually the one which focuses on NC alpha bond. It cleaves along the peptide backbone. So you have very good distribution of your fragment ions. And the problem that I may say is there for triptych peptide when analyzing PTMs by ETD method is that since there is an absorption of an electron, it does not work well with, it needs peptide charge to be greater than 2, which excludes most of the triptych peptides out there. So that brings it more promising technology for more top-down or middle-up approaches, but still the fact that you're not going to lose your PTM still brings it an interesting approach for PTM analysis. So this is a typical spectrum, for example, and you would see that most of the fragments that you see here are coming from the backbone breakage rather than the loss of PTM. Now, this was the most technical part, I would say, in terms of mass spec. Now, assuming that you had the best instrument and you chose the smartest fragmentation method, you used the correct enrichment method, you now have data that is acquired, and now let's see the magic that Jürgen's pipeline does to our data analysis and what are the individual steps there. So this is the pipeline that, again, we've described in this paper, so I encourage you to read about it. This is overall summary of quantification depth that we had covered as we were optimizing this pipeline. So the first step after you've acquired data is score calculation. In principle, this is kind of an extension of Petra's talk where she gave you good details on how one calculates Andromeda score from MS spectrum. How is it different only in case of PTMs is you have to now account for things like neutral losses that we just saw in the fragments in the fragment spectra that you were seeing. So in case of phosphoserine or threonine, for example, there is this neutral loss, so you have to account for that. And if there is a diagnostic peak, which was generated in case of a phosphotyrosine, remember this structure. So you have to say, hey, if there is a phosphotyrosine, I would expect to see this diagnostic peak as well. So you have to define it there, and you calculate the scores with or without loss of these peaks accounted for. And this is a very specific, this is a very general case where you know what to expect, and those standard things are already available in Andromeda's search engine. But who gave the talk? I'm assuming Petra gave the talk on how to set up Andromeda search. If you have a new PTM, or if you're looking for drug-protein interaction, you can do almost something similar here, define your modification of interest. So in this particular case, for example, you can define there is a serine residue on which I'm expecting a neutral loss, but I'm not expecting a diagnostic peak. However, for a tyrosine residue, you do not expect a neutral loss, but you're expecting a diagnostic peak, and you can define its composition. So that's how all these things crosstalk in your search engine. Is that clear, or are there any questions here? Okay. And in case of ETD, the search engine is operating very similar. However, the only difference is that there are no neutral losses. It's working very similar to how MaxQuant addresses this, but you're going to develop this multi-layered scoring approach where you use the so-called restricted plus Y ion, or a complete with 1 Dalton added to them, or plus a Y ion. However, most of our analysis, 90% of the spectra are kind of accounted already by this combination of ions. With this, we would move on to how do you ensure further the quality of your phosphopeptides identified in MaxQuant. So there is this layer of PSM, peptide, and protein level FDRs that are there, but for PTM specifically, we have two additional layers. One is you do a score-based filtering of the modified peptides, and I'll tell you why in a couple of slides. And the other one is this conventional target decoy-based FDR thresholding. So these are two additional layers of filtering that are applied when you're thinking about PTM or phosphopeptide or PTM peptide analysis. How do you define these score filterings? These can be user defined. There are defaults out there that are there sitting in the default settings. So you have to go under global parameters, under identifications. For modified peptides, there is a default score of 40 and the delta score of 6. Now, what is a delta score? Delta score is the score difference between the top hit and the next best sequence distinct hit. So what this would take care of is if your phosphopeptide just made into identification, but actually it was another sequence distinct peptide out there, so we want to make sure that they have at least a score difference of 6 between the two to reliably call it that phosphopeptide. So that's another layer of stringency that we've added in our default identifications. And how did we come to these very specific numbers? Was based on this very large synthetic phosphopeptide reference library where you know what you're measuring and what you're expected to see at the end of it. So this was Bernard Kuster's library where it's a huge synthetic library, and we optimize these score filters and delta scores filters based on those identifications. Now, is that clear? Okay. And mind you again, I'm talking about Phosphos library, so we think this should hold true also for other modifications, but just keep in mind that this has been optimized against a synthetic phosphopeptide library. The scores have been working very well for phospho, but if I were you and I was working on any other modifications, I would like to play around a bit with this and see how does it affect your particular case. Now, coming to the next important step, after you've fragmented your peptides well, you've identified them through Andromeda search, how does MaxQuant localize individual sites on these peptides? So here's an example of a doubly phosphorylated peptide, but there are 3 different serine, threonine, tyrosines that are on this peptide. How do we know where was the modification sitting on this? So let's first go in this dimension where you calculate probability of this particular combination, that is first 2 residues getting phosphorylated, by exponentiating this Andromeda score for this peptide. You go on similar way for the next 2 peptides, and to calculate the localization probability, you come now in this dimension where you add up the probabilities each time you expected this particular residue to be modified and divided by the total probability, so on and so forth. So that's really the formula that goes in localization probability calculation. It's a bit different from what we described earlier, but this is what is in the ongoing current versions of MaxQuant. Is this clear? Cool. So now we're talking about the label-free quantification. It's much easier when you're working with the Silac ratios. You are co-eluting the peptides, and there is no confusion out there that where is the intensity coming from. However, when you talk about modified peptides in a label-free setting, it's much more challenging as a task. I almost remember that when I was starting that project, 60% of the department thought it's not going to work, but it worked in the end because of the, I would say, methods and the computational pipeline that supports it. So you have to take into account how can you choose the best combination of peptides that allow consistent site quantification. By that, what I mean is that you have to make sure that you account for miscleavages, that you're not comparing intensities of a miscleaved peptide in your control and a cleaved peptide in your treatment. And you also have to make sure that you're accounting for similar charge states. So use that combination as you calculate the label-free intensities for your peptides across different conditions. And how do you, it's actually a default option, which is turned on in MaxQuant. Therefore, the golden rule, the default options are the best options. You can define this in global parameters under label-free quantification. If you leave this advanced site intensities option on, this would automatically look for this small subset of consistent peptides that are comparable. Okay. With this, I just show you some more data to convince you that label-free quantification indeed works for phosphopeptide quantification. So if you remember, our workflows involved typical treatment with EGFR or following the mitosis. Across all these 4 conditions using quadruplicate analysis, you see there is a very nice tight quantification of the replicates, and these data make sense with respect to the biology that is expected. So in response to EGF stimulation, you see a consistent increase in EGFR phosphorylation on 2 different sites, and you do expect that the CDK inhibitory phosphorylation has to go down during mitosis, and PLK has to increase during mitosis. So it's working pretty well on individual site level, on replicate level, but it's also working pretty good when you look at the broad picture. Doing the conventional clustering analysis, you do pull out relevant motifs and the pathways that are involved in these processes. What is my personal favorite is this 1D or 2D enrichment plot. If I did not know what data set am I working with, it'll remind me, you were working with the cell cycle. It's to that level that these processes have been optimized, and they tell you what's going on with your data set. Next, occupancy calculation. We realize there have been many questions around in the Google group around this, so I've tried to break down this particular topic now into multiple slides. So number 1, why do you need to calculate occupancies? So let's take a case where you were measuring 3% of your protein, which was phosphorylated, is now 6% phosphorylated. So on a fold change level, this is a 2-fold change. However, the same would be true for a protein that is maybe 30% phosphorylated and is now 60%, right? On a fold change level, both of them are 2-fold changes, but these are completely different things when you think about the stoichiometry of these events. So with this in mind, and assuming that you are as interested as me in calculating occupancies, let's just go, don't think about mass spec for now. How would you calculate it in a stepwise manner? So if these are 3 molecules, total proteins, and you have 1 of them phosphorylated, 2 unphosphorylated in response to stimulus, 2 of them got phosphorylated, 1 remains unphosphorylated, one can calculate proportion of the phospho to unphospho, which would be 1 by 2, 2 by 1 here. And from this number, you can calculate the stoichiometry. So visibly, you can see it's 33%, and here it's 66%. So from proportion to occupancy, it would be proportion divided by 1 plus proportion. So that would give you this number. So it's 0.5 divided by 1.5, so you get 0.33 for this case. Now, this looks very easy if all of your species were very similar and you could just follow this calculation. However, we know this is not what we are measuring. this is a case of an apple to orange comparison, like Marcus says. These are not two same peptide species, so there is an assumption they're flying similarly, and this is not what you typically measure. What we measure is actually fold changes across two different conditions. So that's a fair apple to apple comparison that you make. And now using the same logic, but coming up with a nifty equation where you just put in the ratios of these three species, the modified peptide, unmodified, and the protein there in this equation, which is a brilliant idea that came to Jürgen on how to organize these ratios in this form to get occupancies. So if we stayed with the same example that I showed you before, the modified peptide ratio would be 2, right? Unmodified peptide ratio would be 0.5, protein doesn't change. And this equation, what it does here now is, it says to calculate the fraction of phosphorylated peptide, you just have to subtract the unmodified ratio out of the total protein, and the other way for the denominator if you want to calculate the non-phosphorylated peptide. So just putting the two together, then you get the proportion, which is exactly what you would get if you had the luxury of doing it this way. So with this, you can calculate the stoichiometries. Are we all there? Okay. So with this now possible, however, it comes with the requirement, as you would have noticed, that you have to have a peptide ratio for the modified peptide, unmodified peptide, and you also have to account for the changes on the protein level. So you need to have these three measured in your data set to be able to calculate occupancies. And the second important thing is we are measuring ratios, so we are expecting a change that can be put into these equations, right? But the problem comes when we are choosing the reference samples where the expression is very comparable. That is, you do not see much of a change. So just looking closely into this formula, if there are no changes happening at protein level and also on the modified or unmodified peptide level, what you're doing is 1 minus 1, which is 0, divided by 0. So that's not where you want to go. That's where the error propagation comes into being, just because of the way this formula is organized. So how do we address this? The formula is cool. We now add what we call as an occupancy error estimate to this formula. So the idea is that if we are not seeing much of clearly these changes, you filter out the unreliable occupancies that come up with the very high occupancy error estimate. And this is too much equation for me, but I would just say that, again, you can get all these components of this equation that are defining the occupancy error estimate. And here is the measurement error that is associated with this error estimate. With these two things available to us, and they're again well described in this paper, although in the supplement, so you'll have to go dig deeper into reading those details. What one particular advantage we found with the label-free methods was that you have now multiple reference samples. In SciLac, you are just measuring heavy to light, or medium to heavy, or so on and so forth, and your occupancies are calculated with respect to the reference that you measured your SciLac against. However, in a label-free experiment, if you have 20 different samples, they all can act as a reference to your particular site in one experiment against all the others. So what you can do is really dynamically select, not you, MaxQuant does it already in the background. It dynamically selects the most distant sample for that particular site. So that allows you to see for the combination of the conditions in which you can minimize the error and use that to calculate the occupancies reliably. So that is something which was not there in the algorithm before and was added later. So now talking about all these technical things, how we got these occupancies, got them working, what does it mean for 3 biological conditions that we were analyzing? So here is an occupancy landscape for an untreated HeLa cell. So from 0 to 1, these are really low stoichiometry phosphorylations. These are high. The overall landscape doesn't look much different in case of EGF. However, in mitosis, which is actually almost a phosphoburst associated with many events involved during this process, there's a huge burst of phosphorylation events associated with high occupancy changes. So again, talking in terms of numbers, We could calculate occupancies for more than 16,000 phosphosites, but when we applied these error filtering, it's 50% of them where we could say this is a reliable estimate. Now, there was a very interesting question that Tuna asked me then, like, okay, so you're seeing mitosis samples and you're seeing this very high occupancy. It doesn't look like you need to enrich them to see them, right? And if that was true, we asked, can we actually see high occupancy sites without phosphoenrichment? And actually, while in control and EGF, we found only about 50-ish sites from the proteome runs. There were more than 2,500 sites that we found without enrichment in mitosis, which says that our occupancy landscape change is reflected already in our ability to see these sites without enriching them in the proteome. What I was also very excited about was that now you can not only do the serine and threonines, which have been dominating the field so far because of the tools, we were also able to see, at least for 250 tyrosine sites, how does it look in terms of their occupancies across different conditions. So you see that it's not so high in case of control and mitosis, but when you give a stimulation that works through tyrosine signaling, is a huge change in occupancy landscape. And if you turn off all the tyrosine phosphatases, there is a huge jump in the overall occupancy. So it looks like these phosphatases are really acting like vacuum cleaners for these phosphotyrosines all the time, keeping them not functional. As soon as you remove this switch, it's all going up there. And it's so beautiful for me to see them, at least in this format, reflected in the occupancy landscape. And if you now compare this, the same thing, but keeping in mind the differences between tyrosine and serine threonine, just in the resting conditions, you would see that tyrosines are kept at much lower occupancy than the serine and threonine. That's rested case or unstimulated case. Whereas in mitosis, where serine threonine events are the major players in the signaling pathways, you see that it's reflected in high occupancies. Whereas in EGF, you see that the tyrosine phosphorylation or occupancy changes are much broader, clearer. So where am I going with this is that the controls that are employed in keeping these sites shut down are different in case of serine threonine versus tyrosines, because there's this whole idea of tyrosines being different modification than serine and threonine, and it has much higher level of controls associated with this modification. So the other interesting question for me was extent of protein phosphorylation. So just these numbers are purely based on this particular study, where we had coverage of more than 10,000 proteins, close to 11,000, and more than 50,000 phosphopeptides. And we had class 1, I'll explain that in some time. And we had class 1 phosphosites close to 40,000. Now, back in 2006, when Jesper published this EGF study, we said there are like 2,200 proteins that are phosphorylated. Then in 2010, when the technology got better and our coverage was close to 25K peptides, we said 6,000 proteins are phosphorylated. Now here I come with 50,000 peptides and I say, hey, close to 8,000 proteins are phosphorylated. So actually you can generate this curve with your ability to identify the least abundant peptide. And this shape of this curve should tell you, if I got more sensitive, we are going further and further in terms of saying, I can have evidence for phosphorylation on more proteins than what I know right now. That's why I said the 3/4 of proteome phosphorylated is still at least, it's the current confirmation we have, but looking at this curve looks like we'll have evidence for almost every protein to be phosphorylated. With this, let's get to now the practical aspects of how do you set up search for modifications with MaxQuant. So thanks to this very user-friendly interface developed by Jürgen, it's pretty simple. You go to group-specific parameters. There you go to modifications. You choose your favorite modification. Clearly, my favorite is there. And you bring it on this side, define it under variable modification, and that's it. Now, there was this discussion about why do you need to discard the unmodified peptide as you do the protein quantification, and I thought this would be a good point to talk about it, since you're familiar now with this example, where you see that the modified peptide changes twofold, unmodified reduces to half, whereas the total protein hasn't changed. So actually, you would not want to include this unmodified peptide when you are quantifying the proteins, right? And that's the option that you check, and it's actually default checked when you say discard unmodified counterpart. Now coming to the next favorite topic of discussion that we had 2 days. Now if you were doing a typical proteome and phosphoproteome analysis, what would your experimental design look like as you define it in MaxQuant, and why we do the way we do it here? So if you see, this is an example case of single shot analysis with 3 Silac replicates. You got your 3 phosphophiles, 3 proteome files, and what I'm calling them are similar experiment name if they belong to the same experiment, but I call them different groups. Can somebody tell me why are they called different groups here? Or what would be the advantage here? Absolutely. So if I have enriched for phosphopeptides in only this subset of my data, I do not need to search for this modification when I'm doing normal proteome searches. So I control that by defining them under 2 different groups, right? So in one of the groups where you're doing the proteome searches, you would not define any variable modifications, whereas the other group you would say, hey, do include variable modification in your search. Is that clear? Sounds good. Now, why do I have different fraction numbers for phospho and proteome, but I do have same fraction numbers for different replicates of phospho or proteome? But why this way?
If you have fraction 1 and 2, then it would also match because it also matches.
Exactly. So we do not want to match phospho and proteome samples because they're different. But you do want to match your fractions within, whether if they are proteomes or phosphoproteomes, correct? The last thing, why do you give them the same experiment name? Otherwise, how would MaxQuant know that they are same experiment and would not be able to do any occupancy calculation for you? It has to look where is the information for unmodified peptide, modified peptide, protein changes, right? So if you've combined these 2, I mean in general, not only for occupancy, you need to know that these 2 are same replicates, and then it'll automatically look for what is the modified peptide, its unmodified counterpart, which is most likely sitting in your proteome runs, and the total protein ratios. So the moment you've given it same, you don't need to press a button for occupancy calculation. It happens on its own. Is this clear? Sounds good. Last, this seemingly complicated, but exactly an extension of the last experiment. Now, instead of a single run analysis, you're doing fractionated analysis. So each of your replicate is now fractionated into 3 different fractions. How would you do now? The setting of fractions, groups, and replicates would almost remain the same, just that you want to do fractions as 1 to 3 or 11, 12, 13. Just do not match proteomes and phospho. Is this clear now? Great. So with this, the last point about keeping in mind that when you see a change in modification, that may happen not just because it's changing, but because there's an overall change in the protein level, right? So this is again the example case you'll be seeing. Instead of now 3 molecules where 1 is phosphorylated, we now have 6 molecules where 2 are phosphorylated. If you just followed the fold change on the modified peptide level, you will say, I do see an increase in the modification. However, at the protein level, it's also changing to double the amount that it was. So in this case, it's not the site modification, it's actually total protein expression that has changed. So you need to keep that in account as you see any site changing. Okay, did I finish it before time? Before we take questions, Here is a shameless advertisement. I'm looking for one of bright scientists out here. You all qualify 50% already because you're in the summer school, which means you're clearly interested and you know what you want to learn. I'm looking for a proteomics scientist, PhD level, and if you're interested in doing drug discovery in pharma setting, please, you're welcome to get in touch with me and write me also on this email ID. With this, I'm open to questions, happy to take more.
Hello, over here.
Yeah.
I have actually two questions for you. So
Can I please hear him?
sorry, I have two questions for you. The first one is, does it make sense, or when would it make sense to put a threshold on the localization probability?
So that's going to be part of the tutorial. I'm going to describe all that in detail. So yes, we do handle localization probability in 3 different classes. We call them class 1, 2, 3. However, for me, traditionally in the lab we say anything above 0.75 localization probability, I can reliably trust that. But the others have used 0.9 also, and it doesn't change the number so much. If you see the localization probability distribution, most of the sites indeed are there. So you can cut it off there. And I'm glad you asked this question because when we report the depth of our coverage at the site level, we report only class 1 sites. So you do not just go all the way when your localization probability was 0.2 and you're definitely then overcounting. Okay. However, a good way of talking of the depth of the coverage would be at the phosphopeptide level, because those are the species that you're seeing directly in your samples, and any sequence distinct phosphopeptide is a good measure of the depth of your coverage. Yeah.
Okay, I have a second question, sorry.
Sure.
As actually a curiosity exercise, so we have now increasing number of peptides that we identify in whole proteome, right? It's increasing over and over the years. What would be the limit that we don't need any more enrichment so we can detect the phosphoproteome inside the whole proteome?
Good question. If you remember the talks from first day, you realize, or the talk that Florian gave, this gray border that we are not covering, the gray features that we are not covering compared to what we are seeing. So in my opinion, we are not yet there even with respect to the proteome. Phosphors which are not, which are sub-stoichiometric, or acetylation that Chino was talking about, which is like even really low stoichiometry. I think, to me personally, this is still far. However, that's the overreaching goal of PTM proteomics word, or proteomics word. I can't put a number there. So.
Okay, thank you.
Here. So I want to ask this site occupancy. So I always observe one particular site, there are many species. So how do you deal with it? Like this so the S35 on this peptide, and then it's also on a peptide has double phosphorylation. So how do you
Yeah, so it's not just occupancy, it's also asking how do we center our data from phosphopeptide level to a site level, because you got sometimes miscleaved peptides, you got sometimes different charge states and all. So that's something which is done already in the background of MaxQuant, where it's accounting for the overlaps and adding up the intensities and picking up comparable peptides as you focus on the site level.
So it has its own algorithm?
Yes.
Okay.
So you don't do it as a user. It's the algorithm in the background. Yeah.
Do you ever worry about?
Oh, thank you.
Hi. So by grouping the search for total protein expression versus phospho-enriched samples, do you ever worry that you may miss phosphopeptides that are found in the protein fractions?
I wouldn't worry if I was, like I said, that without enrichment, I don't see much phosphoproteome. If my enrichment has worked reasonable, I am going to capture much more depth. So let me go the other way. So if you're looking at the proteome, what are the chances that you'll see a phosphopeptide depends on how abundant is it. And that's going to be some of my top peptides in the enrichment, so I'm not going to miss them anyway.
Yes. Okay.
Wave
Can
is-
this? Yes. Okay. Here.
Thanks.
Can the site occupancy also deal with, for example, if you have two different kind of modification on a single site? Can it also count for that?
I don't think we are handling that at the moment, though it's an interesting question.
We have another one here.
Sure.
Hi. I have a question about the site occupancy that you explained for SciLec samples. How accurately can we apply it on the label-free quantification? And will the number of proteins identified in the proteome and the phosphoproteome influence this site occupancy calculation in label-free quantification?
Yes. So it's a twofold question. One, whether it works for label-free or not. So the study that I showed you now is actually label-free. So this whole thing was optimized for label-free quantification across those conditions. And you all have that data set with you to play around with on your USB sticks. It does work, and this error occupancy error estimate is calculated with label-free, and we can filter out. So all the data that I showed you was label-free, so it's working very well. The second question was how would you make your sample in a way that you can get occupancies out of most of your data set. So if you realize, I spoke a lot about the trend moving away from single shots, from fractionation to single shots. Now the downside of that is that the chances of you seeing the combination of three ratios that are required for occupancy calculation reduces. You need to have the modified peptide, its unmodified counterpart, which is a very, very specific peptide that has to be seen in your proteome run, right? So if your aim is occupancy, then you better fractionate your proteome pretty well to maximize your chances of seeing that unmodified counterpart. Protein ratios, they are still okay because you'll have enough peptides per protein to get that third component. So to reach the unmodified peptide, you need to fractionate it pretty well.
Hi, here. I wanted to ask, how do you control the reproducibility of the titanium dioxide enrichment in label-free quantification?
Sorry?
Reproducibility of the enrichment on titanium dioxide.
Well, so if it's SciLec, it's already combined, right? When it's label-free, of course, you have to have robust protocols. You cannot say, in one sample I had 5,000, another 50,000. It's not going to work. So to some level, once you start looking at your histograms or once you start exploring your data in Perseus, you know that are these comparable samples to even move forward or not. And I will talk about some of those quality aspects during the tutorial. But it's almost like it's any other scientific experiment. You know that are they comparable experiments to start with or not. But there are estimates on correlations, number of species overlapping or not. So, yeah. Yes.
Just to follow the previous question, if, for example, you do a comparison between a treated sample and non-treated sample, and let's say the treated sample, the cells will grow less, so I have less protein in it. Mm-hmm. And is there any way to normalize the data in a way they can become comparable? Because obviously, what happened is every time I look at my sample, one will have like far more protein than the other one. So I don't know if what I see is because there's less protein because the cells grow less, or if it's because there's an actual difference. So it's really tricky to know how to compare them, because whatever I do my experiment, they still have a response to the drug that changed the protein level.
Okay. So there are two rules that you can usually apply when you're doing this kind of data analysis. Number one is that a stimulus is only going to change a fraction of your signaling events, right? Which means that most of the stuff that you've identified in your control and treatment should overlap. There should be a huge overlap. And the outliers then can be reliably and statistically validated. So that's the right way of doing it. If you do not have good overlap, the statistics are also going to fail. That's one level. Now talking about normalization at the protein level.
Yeah.
Yeah. So So there are two ways of approaching this. Since you've measured the proteome and phospho, and your files are linked in the Perseus output tables, sorry, MaxQuant output tables, you can just call in the protein ratio into your phospho file and normalize it. But you have to, this comes at a cost, that not for each site you may or may not have a protein ratio, right? So in some cases, depending on how the experiment worked and whether you had fractionation or not, you may be even cutting down half of your data, right? That's not very smart move. What I instead do is I work with the entire phospho data on its own, and then I bring my outlier. That's a smaller subset on which then I normalize for the protein or not. So that's most appropriate way of navigating the data analysis. I hope that helps.
So if I understand well, you work with the phospho data first, and then you compare it to a subset of the whole.
So let's say, so I then look, if I'm going to put my money on some of these regulated sites, then I look whether there is a protein quantification available for that or not. But reducing my data to only normalized for protein sites is going to reduce the capability that I have to on the statistical level. So, yeah.
Thank you.
Okay, Les, thanks for an amazing talk. And